Hi everyone, I'm Felix Samuel, a graduate student in the Department of Food and Animal Science. I'm here to present the effect of melatonin on the physiological and behavioral parameters of calves exposed to summer heat stress. The heat stress is a major constraint to animal production, especially during the summer heat period. They redistribute body nutrients, leading to decreased growth rates and productivity. Melatonin is a pleiotropic, multifunctional indolamine derivative, which is produced in the pineal gland and has been documented to have broad-spectrum antioxidant and free radical scavenging properties, among other functions. Temperature, respiratory rates, behavioral parameters, and serum biomarkers can be used to assess the level of stress animals are exposed to. This study, the objectives of this study is to evaluate the effect of melatonin on the physiological and the behavioral parameters of calves exposed to summer heat stress. 20 apparently healthy calves were recruited into this work. They were divided into two groups. The first group included 10 calves treated with melatonin and the other control, which is not treated. 25 milligrams of melatonin were implanted subcutaneously in the treated group once every 28 days for a period of 70 days. Rectal temperature, respiratory rates, hemograms, as well as serum for heat shock protein analysis and growth hormones were analyzed. Okay. The result of this study shows that the thermal heat index obtained during this period were actually above the level reported for the thermoneutral zone of calves in the temperate region. Hemogram shows no significant difference between the treated and the untreated group. Respiratory rate, however, was significantly lower in the melatonin-treated group than in the control group. This could be attributed to the possible amelioration of oxidative damage in the lung parenchyma by the melatonin in the treated group. Temperature also was significantly lower in the melatonin treated group than the control group. This can be due to the function of melatonin in downregulating prostaglandin synthesis, which could upregulate or alter the thermoregulatory center, increasing the thermal core temperature. The average daily gain was also measured. There was numerical increase too, but the increase between the treated and not treated was not significant. The heat shock protein, which is a very sensitive protein that is used to assess the level of stress animals undergo. The heat shock protein was significantly lower in the melatonin treated group than in the control group. The heat shock protein is elaborated during the stress period at a very high level so as to protect the system from oxidative stress. But however, melatonin was able to downregulate or reduce the level of oxidative stress imposed by the summer period, therefore leading to a lower level of heat shock protein in the blood serum. The growth hormones were also measured. However, the growth hormone were not significant in the control and were not significant between the melatonin treated and the control group. The behavioral parameters were also evaluated. However, melatonin was found here to improve behavioral parameters, particularly the truth score and the exit score. In conclusion, melatonin lowered the rectal and the respiratory rates during this summer heat period. Melatonin improved the behavioral parameters of beef calf exposed to summer heat stress. Melatonin also lowers or reduces heat shock protein level in the blood, as well as influence the growth hormone level, which can aid in the growth performance of the calves. Thank you.